Right. So good afternoon, everyone. I'm Christina, and I'm a research student in Dr. Susan Potenin's lab at the Department of Microbiology at Mount Sinai Hospital in Toronto, Canada. And today I will be presenting our assessment of the limit of detection of 4 Candida auris screening procedures, including direct-to-agar, broth-enriched culture, direct PCR, and broth-enriched PCR. So Candida auris is a pathogenic yeast that causes severe infections, and it has been emerging worldwide. Since its first isolation in 2009, there have been over 4,500 cases of C. auris that have been reported in more than 40 countries across all 6 continents. And the reason that this is a cause for concern is because C. auris is very challenging to control. Its ability to colonize on patient skin and persist in the healthcare environment allows it to more easily be transmitted between patients in healthcare settings, leading to nosocomial outbreaks. Additionally, like what the first speaker in the symposium had mentioned, multi-drug resistance is common, which severely limits treatment options, and Candida auris is often misidentified as other yeasts by commonly used biochemical methods and automated instruments used in the clinical lab, so it may go undetected. In order to effectively manage C. auris worldwide, a sensitive, timely, and reliable method of detecting and identifying C. auris is needed. So currently, the CDC has provided several guiding procedures for C. auris screening, including using real-time PCR as a fast and accurate method of detecting C. auris, incubating the specimens in a selective salt or dulcitol broth before plating onto differential media, followed by confirmation of its identification using MALDI-TOF-MS, directly plating the specimens on a selective salt or dulcitol agar, and directly plating onto differential media, followed by identification using MALDI-TOF-MS. Now, while these suggested procedures do provide helpful guidance to clinical labs, there is currently a lack of standardization amongst labs for detecting C. auris. So previously, our group assessed various culture-based and PCR screening procedures to determine the optimal procedures based on their ability to detect C. auris. We assessed 3 culture-based direct-to-agar methods in which specimens were directly plated onto either a selective custom dulcitol agar or onto the novel chrome agar Candida Plus medium, which is a differential chromogenic agar that contains C. auris specific chromogens. We also assessed 3 culture-based auris enrichment broth enriched methods, which entails a 48-hour incubation of specimens in a selective dulcitol auris enrichment broth before plating onto either the Candida chrome agar Candida medium, which is a chromogenic agar that does not contain the C. auris specific chromogens, or onto the novel chrome agar Candida Plus medium that does contain the C. auris specific chromogens. And finally, for the PCR screening procedures, specimens were either directly tested for C. auris using PCR, or they were first incubated for 24 hours in either the auris enrichment broth or brain heart infusion broth before being tested for C. auris using PCR. And through that study, we identified 4 optimal procedures that had the best ability to detect C. auris, and they were a culture-based direct-to-agar method onto the chrome agar Candida Plus at 37 degrees Celsius, a culture-based auris enrichment broth-enriched method onto chrome agar Candida Plus at 37 degrees Celsius, PCR direct from specimen, and PCR from auris enrichment broth-enriched specimens. Our current study aims to determine the limit of detection differences between the optimal culture, broth, and PCR-based methods that we identified in this earlier study. And ultimately, we aim to determine the optimal procedures, taking into account the sensitivity, turnaround time, cost, and ease of implementation. So for our study, we tested 11 C. auris isolates, representative of 3 of the 5 geographic clades of C. auris. And for each isolate, a 0.5 McFarlane and saline was prepared and then inoculated into fresh nasal axillary groin perineum rectal swabs. 6 5-fold dilutions of each spiked specimen was then prepared and then used to test the 4 detection methods. So for the culture-based direct-to-agar method, each diluted sample was directly inoculated onto the chrome agar Candida plus medium. And for PCR direct from specimen method, each diluted sample was directly tested for C. auris using PCR after DNA extraction. For the culture-based broth-enriched method, each diluted sample was first incubated in auris enrichment broth for 48 hours before plating onto the chrome agar Candida plus. And finally, the PCR from broth-enriched specimen method involved testing the auris enrichment broth-enriched samples for C. auris using PCR. The limit of detection of each method was then calculated using 2 tools, the first being a probit analysis from the Public Health Ontario Laboratory, and the second was the Analyze It method validation edition, which is an add-on software for Excel, and both were calculated at a probability of 95%. So this graph summarizes the resulting limit of detection values that were calculated for each screening procedure using both tools. And what we found was that auris enrichment broth-enriched methods were the most sensitive methods. The culture-based broth-enriched method was nearly 200 times more sensitive than direct-to-agar, although the halo effect that's associated with broth-enriched cultures had a similar sensitivity to direct-to-agar. And I'll go over what the halo effect entails in the next slide. For the PCR screening procedures, PCR from broth-enriched specimens was on average 100 times more sensitive than direct PCR. And notably, culture-based broth-enriched methods had the same sensitivity as PCR from broth-enriched cultures. Direct PCR was then the second most sensitive method, being nearly 2 times more sensitive than direct-to-agar. And finally, direct-to-agar was the least sensitive method. So this figure illustrates the varying limit of detection of each culture-based procedure using a representative spiked sample. The red arrows on the figure indicate C. auris growth. And I guess the picture is a little bit hard to see here, but Candida auris grows as light blue colonies with a distinctive blue halo that diffuses into the surrounding agar on the chrome agar Candida plus. And the figure also illustrates how direct-to-agar methods allow for greater breakthrough of background flora compared to the auris enrichment broth method, where C. auris is significantly enriched for. And the last 2 rows of this figure illustrate the halo effect that I mentioned earlier, which is a blue coloration at the site of inoculation on a chrome agar Candida plus, approximately 4 hours after incubation. So in making a decision on which screening procedure to implement in the clinical lab, in addition to the sensitivity of the test, other factors such as cost, turnaround time, breakthrough of other species, and workload in the clinical lab will also need to be considered as well. And this table summarizes and compares these factors for each of the 4 screening procedures. So what we found was that culture-based direct-to-agar methods were the most cost-effective and also most easily implemented into the clinical lab workflow, but it does allow for greater breakthrough of other species, and it is also the least sensitive method. The culture-based broth-enriched screening methods were more sensitive, but requires a greater cost, turnaround time, and also extra broth enrichment steps in the workflow. And finally, PCR screening methods had the shortest turnaround time, but also the greatest cost and workload, without significantly improving sensitivity compared to the culture-based procedures. So to conclude, some of the key findings from our study were that enrichment of specimens in auris enrichment broth increases the sensitivities of both culture-based and PCR screening procedures compared to direct from specimen procedures. As well, we found that culture-based and PCR methods have similar sensitivities. These findings, when taken into account alongside other factors such as cost, turnaround time, and workload, can be used to determine which procedure would be optimal for addressing the different needs in infection prevention and control. For example, both direct-to-agar and broth-enriched cultures may be optimal for being implemented as routine screening procedures, as they have lower relative costs and workload, making them more cost effective and more easily integrated into the existing clinical workflow in order to support the higher volume of screening, while also maintaining similar sensitivities as their PCR-based counterparts. However, for situations that demand more rapid identification, such as in outbreak situations, PCR-based screening methods might be preferred despite the higher cost and workload in exchange for the rapid turnaround times. So ultimately, when it comes to deciding which procedures to implement, it's important for each laboratory to recognize and consider the trade-offs and balances between turnaround time, cost, sensitivity, and workload in order to determine which procedure would be most suitable in meeting its needs. I would like to acknowledge Drs. Kuss, Corvée, and Iro from Public Health Ontario Laboratory for their efforts in the initial validation study, as well as Ceylon for her helpful insights in putting together this presentation. Thank you very much for your attention.
Thank you very much, Christina. Do we have questions for Christina?
Excellent presentation. Thank you very much, Christina. Did you happen to notice any differentiation between sensitivity of the, what would you try, 3 different clades of the CRS? So did you notice any differentiation between that? And secondly, can you explain a little bit about the halo effect that you were witnessing?
Yes, for sure. So we didn't notice any significant differences in sensitivity between the geographic clades. And as for the halo effect, I'm just going to go back to the figure here. So the interesting thing about the halo effect is that when we plate the spiked specimens that contain C. auris onto the Chrome-agar Candida plus medium, and then we put it in the incubator at 37 degrees for approximately 4 hours, there will be a blue coloration on the agar. So it's not actual C. auris growth. Rather, it's kind of like a blue coloration in the agar itself, very similar to the blue halo around the actual C. auris colony once the colonies actually form. And so the interesting thing about this kind of feature is that there's potential for identifying C. auris in samples before the full 24-hour kind of incubation, which is like the middle 2 rows shown there. The only thing that we noticed is that this halo effect wasn't consistent for all C. auris isolates. So certain isolates would exhibit this halo effect and have this blue coloration, while others didn't. So it's not, it doesn't apply to all isolates, but it is a potential way to alert the lab that perhaps this may contain C. auris because of that blue halo at the inoculation site. And hopefully that answered your question.
Any other questions?